Today I'd like to refresh your memory on the Calvin cycle. And then we need to think a little bit more about the PCO cycle. So the big pie chart here showing the carboxylation, reduction, regeneration. And we have the first phase where that very, very unique enzyme, Rubisco, is participating. So we're going to talk more about Rubisco. So if everything is going okay with the Calvin cycle, Rubisco will take in CO2 and use that to react it with RUBP. And so then you end up with those two PGA molecules by the end of that. And then reduction is getting the PGA to G3P. And for that, you need both a phosphorylation event, so ATP is needed to phosphorylate, and then you need NADPH to donate off some electrons for that as well. And so going from PGA to G3P is the so-called reduction phase of the Calvin cycle. And then the G3P is kind of like a branch point, because most of the G3P will get used in regeneration of RUBP, but then a little bit of G3P can get siphoned off for sucrose synthesis. And if it's getting sent off for sucrose production, that has to happen over in the cytoplasm or the cytosol, okay? So all of the Calvin cycle is happening in the stroma of the chloroplast. So we're still zooming into the chloroplast. So to go from G3P to the RUBP, you also see that phosphorylation is necessary. That's where we did the so-called numbers game of looking at the combining of the G3P molecules. So those are each 3 carbons. And so you combine 2 of them together, that gets you to 6 carbons, the fructose 6-phosphate. And then you take that 6 carbon and combine it with another 3 carbons. So that's 9. They're not showing that molecule because it breaks quickly into a 5 carbon and a 4 carbon. So 5 is the magic number we're looking for. So that one is pretty good to go, but that 4 carbon's not there yet. So if you take that and combine it with another 3 carbon G3P, now you're up to 7. So then again, if you can get up to 10, that's good, because you can split that in half to 2 5s. And so that's what you see happening. It reacts with another G3P to get it to 10 carbons, and then it splits into a 5 carbon ribose phosphate and a 5 carbon xylulose. So the 2 xylulose 5-phosphate, the ribose 5-phosphate, they rearrange into ribulose 5-phosphate. And then you finally see the ATP, because we need 1 more phosphate, because RUBP has 2 phosphates. So you have to get the ATP to donate off a phosphate group, and then you're back up to the ribulose 1,5-bisphosphate. So remember, with the regeneration phase, I'm not asking for enzyme names or names of the compounds even, other than this last step. The very last step, I want you to know that you take the ribulose 5-phosphate, you add in ATP, and your product is RUBP. Other than that, you can just describe it to me by using the numbers game. And so I posted that second set of PowerPoint slides into Educat, where I think I actually call it the numbers game. So just look at that for reference. And then of course, the carboxylation and the reduction, I do want you to know what's going on there. Rubisco is really the only enzyme I'm asking you to know. Okay, so that's what's kicking off the very first part of the Calvin cycle. And so, but knowing the substrates and products of carboxylation and the reduction. Regeneration, it's just mostly numbers game until you get to the very last step. Then I want you to know that as well. Questions on the Calvin cycle or the C3 cycle. So this is the carbon fixation idea that they talk about. When they say it's carbon fixation, they're getting at the idea that you have the CO2 coming into the plant. And actually the Calvin cycle, it's some of that's going to go off for sucrose synthesis. That's really the carbon fixation idea. You're finally making sucrose from carbon dioxide. So some of that G3P is getting exported. What they mean by that export, they mean out of the chloroplast into the cytoplasm. And then will be made into sucrose. And then eventually that gets dumped into the vein, into the phloem, sieve tube elements, and gets sent around to the different parts of the plant body that need it. Okay. Then we said, well, Rubisco, we talked a little bit about how some of the enzymes, 4 enzymes, and also Rubisco of the C3 cycle are light activated. So this C3 cycle or the Calvin cycle does not run at night. Then we started talking about Rubisco being a little bit sloppy, where it doesn't have a real tight binding affinity or a high binding affinity for CO2. It actually has a carboxylase function, and it also has an oxygenase function. So the CO on Rubisco stands for that. And so if it's going to see more oxygen, it's just a kind of a competitive thing. Whatever's in higher concentrations around Rubisco, that's what's going to come into the active site. So if for some reason there's low levels of CO2 around Rubisco and still plenty of oxygen, then oxygen wins and comes into the active site. And so now you see what it's doing here. It's reacting with RUBP, and you do get one PGA that can immediately go in to the rest of the Calvin cycle. But that phosphoglycolate, it's only 2 carbons. There's no enzymes in the Calvin cycle that can immediately act on that and create PGA out of it. And so what we see, the goal here is when you look at the PCO cycle, it's really, you got to get that 2-carbon phosphoglycolate converted to a PGA so it can then enter into the Calvin cycle. But as we saw last time, that's a production. So we were looking at this last time. I think this is pretty much where we left off. So for the PCO cycle, you can see there's 3 different organelles, and I need you to know what are the organelles involved, okay? So it kicks off in the chloroplast, because that's where Rubisco is, of course. And then you see at the top left here, the RUBP and oxygen reacting. And so the products are just like what we saw on the previous slide. One of the products is phosphoglycerate, so that's swooping off to the right here. And then I typed in or drew in the C3 cycle. All right, so that PGA can immediately get fed into the C3 cycle that's cooking along in the chloroplast. But that other product, the phosphoglycolate, that has to go through all of this, and it actually does it twice. You have to get 2 runs of the PCO cycle to get enough carbon to build another PGA, because this is just a 2-carbon. So as we work through this, I want you to know the order of, which organelles and the flow. So what you see here is the chloroplast, the products from the chloroplast are feeding into the peroxisome. And so what you see here is phosphoglycolate getting converted to a glycolate. That's the one that's going to mobilize out of the chloroplast. It has to find the peroxisome, get in, and find the enzymes to convert it to glyoxylate and then to glycine. So what I'd like you to know is, what are the products that are leaving each organelle? Okay, so in this case, we would say glycolate is leaving the chloroplast and entering the peroxisome. And then glycine is the product in there that will leave the peroxisome and come into the mitochondria. Then what you see happening in the mitochondria, what they're trying to show here in this drawing, and I'll show you a separate drawing where they do it a little differently, and hopefully one of these will click with you. But they show glycine leaving the peroxisome and coming into the mitochondria. But then if you look over here on the right side of the mitochondria, you see another glycine showing up right over here on the right side. What they're trying to say by this is that you have to have another glycine coming in. So essentially what they're saying is the whole left side of the PCO cycle has to run twice. You need 2 glycines in the mitochondria because that will create enough carbons to do the next reaction. So it's kind of a carbon counting issue here. And so this front half of the PCO cycle has to run twice. You end up with 2 glycines in the mitochondria, and they run through this reaction that ultimately leads you with the product serine. Okay, now you're at 3 carbons. Serine is 3 carbons. So, but something else is going on in the mitochondria that's of particular importance for the plant. You also see going on in here this CO2 molecule. So CO2 in the lower left here of the figure, what they're saying here is carbon dioxide is one of the products that's cooking along here in the mitochondria. When glycines are coming in, CO2 is one of the products, and it gets respired off by the plant. So in what they say is it's lost to the plant in the sense of they can't use that CO2 because over in the mitochondria, it just goes out and it doesn't find its way back into the chloroplast for fixation. So they call it a loss of carbon. It's respired off. Very, like we're respiring CO2 right now. We're losing carbon. And so they say that CO2 is a loss to the plant. So that's a definite negative for the PCO cycle. You don't want, that's exact opposite of what the C3 or Calvin cycle is trying to do. Trying to take the CO2 and fix it. Here we're losing it. So that's a big deal. And that's happening in the mitochondria. So when these 2 glycines come into the mitochondria and react, the ultimate product is serine and CO2. Those are the 2 important ones. And then the serine has to mobilize out of the mitochondria, find the peroxisome, goes through a reaction, where you end up with glycerate. Well, now we're getting closer. Phosphoglycerate, we're getting close. So now glycerate moves back into the chloroplast, and now you see the input of energy. You need ATP to phosphorylate it so that you end up with phosphoglycerate. So when you look globally at the PCO cycle and say, what is this whole thing accomplishing? It's taking that phosphoglycolate and running a series of reactions where you end up with phosphoglycerate. And phosphoglycerate can then immediately feed into the Calvin cycle. But in order to get that phosphoglycerate, you had to expend extra energy. ATP is needed. You lost some carbon in the form of carbon dioxide. And it's complicated. This is kind of a complicated process. You have 3 different organelles. You have to imagine these molecules having to shift, going from place to place and finding the right enzymes, and it's time consuming and complicated. Here's another view of it. I think this is the one in your text. Here they show 2 glycines. They just put 2 glycines in the mitochondria. But I like this one for you to look at just to convince yourself of the carbon counting. Okay, so phosphoglycolate is 2 carbons. And so you see 2 carbons all the way. So then if you add 2 glycines together, well, that's 4 carbons. But remember, one of those carbons gets lost in the form of CO2. So when you get to serine, you're to 3 carbons. All right, and now that's good because we know PGA is a 3 carbon. So it's like, okay, at least now we have 3 carbons. So now you see the movement back into the chloroplast. Eventually you'll have glycerate shifting into the chloroplast, and that's really close because you just add a phosphate group and then you have your phosphoglycerate produced. So I like this one just to convince you of like the carbon counting so you can understand why you need 2 glycines to begin with. All right, so they say that the PCO cycle essentially has to run twice. It's really the front half of it because you need 2 glycines to make sure that you can end up with 1 phosphoglycerate. So that's putting them side by side. Questions? on the PCO cycle. Clear as mud? All right, so the way they phrase it, they say for each 2 turns, it's really the, the front half of it has to run twice. You get 4 carbons from that, because that's 2 glycines, that's 4 carbons. And from that, you'll end up with a PGA, which is good, but you'll lose a carbon as CO2, which they don't consider very good when you're actually the plant's trying to grab CO2 and fix it. So it's kind of the opposite of what the Calvin cycle is doing. So basically what you say is 75, 3 out of the 4 carbons is recovered through the PCO cycle. So they say 75% or 3 out of 4 carbons are recovered and put back into the C3 cycle, but then that last carbon is given off in the form of CO2. So that's why they call it photorespiration. It's respired off. So 3 out of the 4, that ends up as a PGA, and then that can immediately feed into the C3 cycle. It's there in the chloroplast where it needs to be, where the C3 cycle is cooking along. But unfortunately, that CO2 given off in the mitochondria seems to be a complete loss. So of course, plant physiologists have been scratching their heads for decades about this and getting into lively debate about why is Rubisco so sloppy? Why is it such a sloppy enzyme? Why didn't it evolve to be a very high binding affinity for just CO2? And so that has been a conundrum that has plagued plant physiologists for quite some time. If you just look at some of the, reasons why, if you think about some of these ideas. So when you look at gases dissolved in water at 25 degrees Celsius, the ratio of gases of CO2 to oxygen is about 3 to 1. So what that's saying is there's more CO2 dissolved in an aqueous solution. So CO2 is a little easier to get into solution than oxygen. So, we're thinking about our atmosphere and thinking about the gases coming and dissolving into the plant's cytoplasm. Well, if inside the plant, where, you're thinking about the plant cells, the cytoplasm, the chloroplast, if you have a situation where if oxygen levels are low and CO2 is high, that's favoring the Calvin cycle or the C3 cycle. It's favoring carboxylation, where CO2 reacts with RUBP. So when they say carboxylation, that's what they're saying. They're saying RUBP and CO2 are going to react. So that's the start of the Calvin cycle. So you have to keep the oxygen levels low enough and then have enough CO2, of course, to allow for the Calvin cycle. But if you have a situation where CO2 levels drop for some reason and O2 levels rise comparatively, remember it's a competitive thing. So if there's more oxygen than CO2 around Rubisco, then oxygen wins, comes into the active site. And then you have the PCO cycle. So oxygenation is just saying that Rubisco will let oxygen in to its active site and react it with RUBP. So under normal kind of standard conditions, CO2 is favored because of the ratio here. You have more CO2 in an aqueous solution. So in the cytoplasm, you should have more CO2, but you have to remember that the plant is dynamic in its ability to open the stomata, close the stomata. How much oxygen is it giving off through photosynthesis? And so you have all those factors to consider, and that can definitely play with the ratio, where you might end up where you have low CO2, higher oxygen. Another thing to keep in mind is hotter temps around the plant the enzyme will favor oxygenation. So tropical plants, desert plants, they are in an environment that actually for long stretches of time will favor the PCO cycle, because of the hotter temps. And remember, if it's 80 degrees around the plant, you have to think about the plant leaves. They're going to be hotter than 80 degrees because of the pigmentation is absorbing that heat energy. So if you take the temperature of a leaf compared to the temperature of surrounding air, the leaves are always a little bit warmer. So keep that in mind too. So increasing the temperature, so now we're thinking about tropical plants, desert plants. They are at higher risk for sure of the PCO cycle running more continuously, which is not a good thing. Another thing, why is that the case? When the solubility, SOL is solubility, the solubility of gases decreases in general, any gas, the solubility will decrease as you warm that solution. However, oxygen is less affected. So what that means is that oxygen stays in solution longer than CO2 under high temps. Okay, so that means oxygen stays dissolved longer. So that means Rubisco sees more oxygen at higher temps. And so then you get the PCO cycle. So these several bullet points are pointing to the idea that, if you're a plant that lives in consistently warm conditions or consistently hot conditions, especially when you want to open your stomata or even close your stomata, really, we'll get into that, you're at a higher risk of running the PCO. And they've done head-to-head studies where if you force a plant experimentally to run PCO a lot, they suffer in terms of they don't grow as well, their overall biomass is much lower than the control plant that was not forced to run the PCO. So then they also think about Rubisco, like, okay, Rubisco's been around since the first photosynthetic organism existed. And it really wasn't plants, it was bacteria that first did photosynthesis. And there's still plenty of them out there currently, but they're the ones who first came up with, evolved the idea of photosynthesis and had Rubisco to do that. So when you think back to about 3 billion years ago, there wasn't oxygen in our atmosphere. It wasn't there. And so there was no selection pressure for Rubisco to tell the difference between CO2 and O2. Back then, there was no O2. And so evolutionary biologists say, there was no selection pressure for Rubisco to firm up, I just want CO2 because I don't want this O2. There was no O2. And so Rubisco never had to worry about it. And then slowly over millions of years, our oxygen levels arose, kept going up to where they are currently. And so they were saying, just because of the conditions of Earth back then, Rubisco never really had to become with a very high binding affinity just for CO2. Didn't have O2 to even think about, so to speak. When you look at the PCO cycle, there are a lot of intermediate molecules in the pathway, and we're not going to get into it, and you're not expected to know those intermediates in any detail other than just who's leaving and going into the next organelle. But they do see that some of those intermediates feed off into other physiological pathways, and maybe that's important. Maybe that is important that some of these intermediates are made. Maybe they are critical in some way for the plant. So they're investigating that. Some physiologists have said, well, maybe it acts as some kind of safety valve to get rid of ATP. This one I don't agree with, but you do see it every once in a while in readings. They say it acts like a safety valve. And to me, that does not make much sense. So just be aware that some are thinking that way. I don't think it makes sense. Excess ATP. Living organisms don't really have excess ATP. It gets made, it gets used. It gets made, it gets used. So I have a hard time buying that one, but sometimes you see that in readings. Okay. So they're still investigating that. And there's been some recent publications on Rubisco where like the title of the paper is something to the effect of Rubisco's not that bad after all. And, you know, its sloppiness is not such a big deal. And so there's been some interesting new ways of testing Rubisco and thinking about, you know, why is it still sloppy after all these millions of years? It never tightened up to just accept CO2. And so that begs the question, there's probably a reason for that. And so they're seriously looking at that. But so far, there's no solid, like everyone's agreeing to why that is. So, but there's some good data coming out there to think about. But we can't talk about that right now. We don't have time for that. So we're going to get into, okay, if you're a plant that's in an environment that's consistently warm or hot, you do run the risk, a higher risk of a PCO cycle running. So what we've seen is that plants have evolved mechanisms to deal with this. And one is called the C4 cycle. And then we call the plants that do this the C4 plants. Now, remember, every single plant does the C3 cycle. So C4 plants do the C3 cycle. It's just they have another cycle added on top of it that creates a situation that suppresses the PCO cycle. So it greatly diminishes the PCO cycle in these plants. So we're usually thinking about plants that are native to tropical and subtropical environments. Those are the ones that tend to run the C4 cycle. When you look at these plants under the microscope, like we do in BI 230, they look different. Their leaf anatomy is different than a typical straight ahead C3 plant. And so what we see is that they have a very well-defined bundle sheath, which is a ring of cells that surround the vein. I'll show you some pictures. So they have a very well-defined, very plump ring of cells called the bundle sheath that's right around the vein. And then the mesophyll cells, their design is their an arrangement in the leaf is a little different than what you see with a C3 plant. I'll show you some pictures. C4 plants, what they end up doing really when you look at how the C4 cycle works, they spatially segregate where certain parts of the pathway happen. And the benefit of that is that the Rubisco enzyme gets kind of segregated or sequestered away from oxygen. And so they have a physical or a spatial way to kind of split up where certain reactions are happening physically in the leaf. And the big picture of that is it creates a situation where Rubisco really doesn't see much oxygen. So therefore it won't run the PCO cycle, and it just keeps running Calvin cycle, Calvin cycle, Calvin cycle. So it's really kind of an elegant, simple solution. Just physically separate some of these pathways. So don't forget though, every plant has a C3. I like to think of the C4 cycle as kind of tacked on the front end of the C3 cycle. Kind of links into the front end of it. So I'll show you what I mean. Then if you examine these plants that are running the C4 cycle and you say, what's the payoff to this? Is there a payoff? Yes. They are able to thrive in drought conditions compared to a straight ahead C3 plant. They do much better under drought. If they are experiencing any level of water stress, they do better than C3 plants under those same conditions. If you measure their photosynthetic rate, they can cook along at a higher rate than straight C3 plants. So that means if they're doing photosynthesis more efficiently and rapidly, that means they're growing faster, they're producing more yield. And if you're a grower, that's a big deal. If you're trying to harvest these plants for food production or whatever, you want some good yield from that. So depending on the plant species, they say roughly 2 to 3% photosynthetic rate compared to plants in the same environment just doing C3. So really the C4 cycle, it's one of the things that it accomplishes is it concentrates CO2 right around rubisco. So again, it's forcing the situation to make sure that rubisco sees lots of CO2, lots of carbon dioxide, very little oxygen. And if it can keep doing that, then remember, it's a competitive thing. Rubisco is just going to keep taking CO2 in. If there's more CO2 around rubisco, that's what wins and comes in. And of course, that allows for the Calvin cycle, Calvin cycle, Calvin cycle. Keeps running along all day. All right, let's look at some leaf anatomy. So this is the cross section of, I believe this is maize or corn. Doesn't really say in here, but I'm pretty sure that's what they went for. Okay, so what do we got? So this is a cross section. And what you're looking at are veins. Here's the major vein, I believe, right in the middle of this image, and the minor veins on either side. Okay, so the major vein and then the minor veins. One thing, especially on the minor veins, it's really obvious. You see these big, fat, plump cells that are surrounding the vein. So the xylem are reddish, the phloem are those more delicate looking blue-green, and then that ring of cells around the vein, that's your bundle sheath. And you can see around the central vein, around the minor veins, the bundle sheath is very obvious. They're big and plump. Also, you see that the mesophyll cells are pretty much just all spongy mesophyll. You don't see the palisade mesophyll or parenchyma, or those column-looking cells. You don't really see those. It's just pretty much all spongy parenchyma or spongy mesophyll. All of the kind of dark green blobs that you're seeing in this image that are in the bundle sheath, that's lots of chloroplasts. That's another dead giveaway that you're looking at some kind of C4 plant. You see lots of chloroplasts inside the bundle sheaths. Here's another image. And again, this one's really nice. You can see the really plump bundle sheath cells surrounding the veins, really obvious. And then the chloroplasts in there as well. And then you can see the parenchyma or mesophyll cells. They're just all spongy. There's no palisade in there. Here they're looking at a C3 where it's not as dramatic with the bundle sheath. There's not as many chloroplasts. Here is a C4. They've actually stained the chloroplasts pretty well here. Oh, one of the other things I wanted to point out. And they kind of show it. Kind of showing it here where the spongy parenchyma cells, they kind of hug close to the bundle sheath. They're physically very close. And they tend to see that. It's a little hard to see in some of these preps. And I kind of see it here. You can see the spongy is kind of preferentially hugging up to the bundle sheath. And you see kind of a dead airspace there away from it. C3, it's not so obvious with the spongy hugging the bundle sheath. And then the drawing here, they're trying to show that as well. So the C4 leaf is up top here, the top left. So the way they drew it, they're trying to show how the spongy mesophyll are hugging up tight to the bundle sheath. But here in the lower right, an example of a C3 leaf where you tend to see very defined palisade cells, and then the spongy are more scattered. They don't hug up against the bundle sheath so much. So the reason that they hug up like that is that we see the spongy mesophyll in the bundle sheath. Some reactions happen in the bundle sheath, and then the products get sent to the mesophyll. Some reactions happen there, and then the products get sent back to the bundle sheath. And so you need to have the nice close physical connection. You need to have lots of plasmodesmata This next image is trying to show the plasmodesmata, all the holes. Those are the tunnels between the bundle sheath and the spongy parenchyma. So lots of communication and flow of chemicals between the two. So here they've done a drawing of it where they show the spongy parenchyma or mesophyll cell and then the bundle sheath. And so you can see that you have a spatial segregation. So one of the ideas there is that the artist here drew the Calvin cycle in the bundle sheath, but you don't see anywhere did the artist draw a Calvin cycle in the spongy parenchyma. All right, so it's spatially separated. So most C3 plants, every single leaf cell does the Calvin cycle. But in a C4 plant, it's like, nope, we're going to keep at least most of the Calvin cycle happens in the bundle sheaths, the vast majority of it. All right, so that's a big difference. Okay, so with C4 plants, the C4 cycle actually kicks off in the mesophyll cell, where they'll bring in CO2 into the spongy mesophyll. And it's not rubisco that's going to interact with it first. All right, so what's happening is the C4 cycle, instead of having rubisco play a role in that, you have you don't have RUBP either. You have phosphoenolpyruvate. So CO2 comes into the mesophyll or the spongy parenchyma cell, reacts with phosphoenolpyruvate. Another way to express that is PEP. You can just write the abbreviation. So here you see the HCO3 minus. That's when you dissolve CO2 in water, it becomes HCO3 minus. It's going to react with PEP, and you get 4 carbon molecules. So that's why they call it the C4 cycle. The first stable molecule is 4 carbons. And then they show after that event that one will mobilize into the bundle sheath. And then a CO2 gets cleaved off of it and fed right into the Calvin cycle. So in the bundle sheath, that's where you finally see some CO2 getting delivered to the Calvin cycle, but it's really the C4 acid that is delivering the CO2 into the Calvin cycle. Once that CO2 gets delivered, then another product of that reaction is a C3 acid that gets shunted back into the mesophyll, and then you have to make more PEP to keep it all going. So then the regeneration phase is to get more PEP. So this, for a C4 cycle, when you combine the C4 cycle to the C3 cycle, you have a carboxylation event, but it's very different because you don't have CO2 and RUBP immediately reacting. You have CO2 and PEP, that's your carboxylation. Then over in the bundle sheath, you have a decarboxylation event. That just means that's where CO2 is getting cleaved off and fed to rubisco. And then you have to get more PEP, so they have to regenerate that. So that your last step is regeneration. So it's a cycle. You got to keep it going around and around and around. So let's look at this in a little bit more detail. So now some of the words. So here you have the C4 cycle. The CO2 up top is coming into the mesophyll or parenchyma cell. It's reacting with phosphoenolpyruvate, PEP. And the enzyme, I need you to know the enzyme. It's not rubisco. It's called pepcase. So it's PEP carboxylase. The case stands for carboxylase. So the beauty of this enzyme is it's so picky. It's like, I'm only taking CO2. That's all I'll accept. I'll accept CO2 and PEP in my active site, no oxygen. So it has a nice tight binding affinity for carbon dioxide. When those two, when the carbon dioxide and PEP react, I'm going to teach you about the malate type. There's a few different types of the C4 cycle. The most common one is the malate type, so that's the one I'm focusing on. So the product, the 4 carbon molecule, is malate. Another way to express that is malic acid. So it's a definite acid, okay? So malic acid now, I like to think of it as a vector. If you've learned about vectors in biology, like the mosquito carrying the malaria, we say the mosquito is the vector for the malaria pathogen. And so this is essentially what malate's doing. Instead of a mosquito, it's malate that is delivering, instead of malaria, CO2. So its job is to, CO2 is part of the malate molecule. It will easily, it's much easier for malate, an organic acid, to move in, dissolved in the cytoplasm than it is for just straight ahead CO2 to do this. So it's just a very nice, efficient way. Malate can get transferred easily into the bundle C cell, and then like the mosquito biting the human and delivering the pathogen. In this case, though, malate will get a carbon dioxide cleaved off of it and fed right into the Calvin cycle. So you have to imagine now Rubisco, right where that CO2 is. You have to imagine Rubisco sitting there going, okay, I'll take the CO2. And so the CO2 gets fed to Rubisco over and over and over. As long as the cycle is continuing to run, it floods Rubisco with CO2. And that's what you want, because then the PCO cycle doesn't have a chance to get going. So that's perfect. So then CO2 is coming in, getting cleaned up, all the malate coming in. Well, once you get CO2 off of malate, the other product is a 3-carbon, which is pyruvate. So now pyruvate's job is to find the mesophyll cell again so that it can get regenerated into phosphoenopyruvate. So carboxylation in the mesophyll with that very special pepcase enzyme. Decarboxylation just means malate's getting a CO2 cleaved off of it, and then regeneration. So I do want you to know those 3 terms of the C4 cycle. And then the idea that most of the Calvin cycle is happening in the bundle sheath. It's not happening in the mesophyll, the spongy mesophyll. Questions on that? Good question. They can perform it. It can kick in. If things go wonky, it can kick in. So for example, it takes them a lot longer for their PCO cycle to kick in because they just have such a good way of concentrating. But you could imagine where even with C4 plants, there might come a time during the, maybe even in early morning, where they're experiencing drought and they're like, uh-oh, I can't pull up any water. I'm getting drier and drier. I have to shut my stomata to deal with that because, of course, if living organisms don't have enough water, we all die. So they'll make the sacrifice even during the day and close their stomata. Well, now they have no CO2 coming in. So all this grinds down to a much slower rate, and eventually it might stop. And then the PCO cycle. So, yeah, it's a problem. So a lot of this is driven by how much water is in the soil. Are they experiencing drought? If they're well watered, they'll easily be able to suppress this, the PCO. And if you'd run head-to-head experiments, C4 plants and C3 plants under drought conditions, C4 plants still win. They're still better at it than just straight ahead C3 plants. They're better at photosynthesis and growing better. Okay, so what we just looked at is visual. I just want to write it down in words in case you like to look at it that way. So they call it the C4 cycle because it's the 4-carbon molecule. The actual first stable molecule is oxaloacetate, not malate. I don't even know if they show that. Did they show that on this? No, they skipped that step. So just be aware. And then oxaloacetate rearranges to malate. So really it's CO2 and PEP reacting and you get oxaloacetate. Another way to, OAA stands for oxaloacetic acid, which is another way to say that, oxaloacetic acid. And then that rearranges into malate. And so the PEP carboxylase enzyme runs that reaction and it's very picky. It only likes PEP and CO2. So the fancy way to say that is it has a high binding affinity for CO2, doesn't care about oxygen. And another thing they report when they do the kinetics of the enzyme, they say that it has it's saturated nearly always, which just means that it's very, very efficient at taking in CO2, It's just like really good at it. And so CO2 and PEP reacts, boom, CO2 PEP reacts. So it's very, very active, very efficient. They've done studies, careful lab studies, oxygen just doesn't come in to the PEPcase active site. It doesn't work. So that's really good. So that very, very first step of the C4 cycle is pretty sweet because oxygen isn't a problem. That PEPcase enzyme is very picky. And so once you get that OAA made, it rearranges into malate, and then that's kind of like the mosquito. It will move as a vector. It's delivering delivering the CO2 into the bundle sheet because that's where the Calvin cycle is. Okay, so let's see. This next slide shows the details of oxaloacetate showing up first and then malate. So I'll show you that. So here you have the CO2 again up top on the image coming in and reacting with that 3-carbon PEP with the help of PEP carboxylase, and you get that 4-carbon oxaloacetic acid. And then you do see reducing molecules are needed, so NADPH, so that's coming from the light reactions. So there's a cost. The C4 cycle doesn't come for free, and we'll talk about that. So here you see NADPH involved to get you malate, and then malate moves in to the bundle sheath. The CO2 gets cleaved off and delivered to Rubisco. The other product, pyruvate, has to go back. And then the other big idea here is now you're seeing an input of ATP. So the ATP that you're seeing here and the NADPH, that's all coming from the light reactions. So this is not a free lunch. The C4 cycle, there's a cost to this in terms of energy and reducing molecules. So, however, they've done really careful lab studies. The benefit outweighs the cost. I mean, plants are still doing this. It allows them to adapt and thrive in areas that are consistently hot. And they can deal with drought much better than straight C3 plants. But it's a cost. So, okay, let's do it in words again. So here you have the malate, oxaloacetate rearranges, gets reduced, malate that mobilizes into the bundle sheet, and CO2 gets cleaved off, and that gets fed into the C3 or the Calvin cycle. And then the pyruvate has to go back into the mesophyll cell so that it can get regenerated. Okay, so this is the decarboxylation step in words. What you see here, decarboxylation just means you're cleaving CO2 off malate and feeding that into the Calvin cycle. And then the regeneration is to get that pyruvate to regenerate into PEP that's going to take ATP. You got to phosphorylate it, and it's a funky enzyme. You don't need to know the name of the enzyme. I put it here because some people have asked. I'm not going to ask you to memorize it, but in case you're curious, it is a very unique enzyme. When you add it all up, what they say is it takes 2 extra ATP per each CO2. And then don't forget about the reducing molecules. So if you want to fix 1 CO2 molecule in a C4 plant, it's a total of, when you add up the Calvin cycle and the C4 cycle, it's 5 ATP, 2 NADPH. So that's more than what you'd see if you just zoom in on the Calvin cycle. So that's why they say it's an energy cost. It does cost the plant, but like I said, the benefits outweigh the cost if in those plants that are adapted to those particular consistently warm habitats. So the current push in some research labs right now is to try to get C3 plants to do C4. They're trying to engineer plants that just do C3 to make them do C4. And they're making progress. I think in your lifetime you'll see it, but it's complicated. You know, that's a big pathway to consider. And so they've made some partial progress on it. Last time when I went to a conference pre-COVID, right before COVID, I went to some of the seminars on that, and they are making progress. So some of the, of course, the agricultural crops is what they're interested in. So, and speaking of ag crops, so corn is famous as being a C4 plant. All right, and yet, excuse me, we grow corn up here. You know, there's corn fields up here, but they're in an environment that they're not native to up here. So C3 plants and C4 plants up here, C3 plants actually tend to do better up here because that's what they're adapted to, this type of habitat. You grab corn that's adapted to tropical areas and put them up here, they actually can struggle sometimes up here. So you still have those fields, but okay, this is a table trying to show the, oh, we're not out of time. Trying to show, sorry, we're trying to show where everything is happening. So we'll get into this. I'll review the C4 cycle on Monday, and then there's a couple of nuances here I need to explain. Okay, have a good weekend.